I'm delighted today to welcome Professor Paul Griffiths, who's Emeritus Professor of Virology at University College London, as this year's winner of the Unilever Colworth Prize lecture. The prize, sponsored by Unilever's Safety Environmental Assurance Centre, is awarded annually to an individual who has demonstrated an outstanding contribution to translational microbiology. A truly worthy winner of this award, Paul's work concerns Cytomegalovirus infection. For the UK Department of Health, Paul has been a member of the Joint Committee on Vaccination and Immunisation and currently chairs the National Certification Committee for Polio Eradication, the UK National Certification Committee for Measles and Rubella, and the proposed UK National Authority for Containment of Polioviruses. Today he's going to talk to us about cytomegalovirus, direct and indirect clinical effects and prospects for control by immunisation. Welcome, Paul.
Well, thank you for that generous introduction. My title is CMV Direct and Indirect Clinical Effects and Prospects for Control, and I'm going to spend 2 slides just defining those words. So first of all, CMV is a common infection. 60% of adults in the UK or the USA are IgG positive, seropositive, and 90% in developing countries. And the prevalence increases with age. Most of those people have no symptoms. Those who have antibodies, it's associated with low socioeconomic status and non-Caucasian race. And girls born into families who migrate from high prevalence countries take up the prevalence of their adopted country. So if most people have no symptoms, does it matter? Well, yes, because CMV is a recognized pathogen. It's the commonest cause of congenital infection in humans. It's the commonest, the most serious opportunistic pathogen in transplant patients. And it's a major problem for HIV patients who don't have access to antiretroviral therapy. In each of those cases, end organ diseases require that a high viral load is present to damage the tissues. It's called retinitis and pneumonitis, etc. And the virus can be detected histopathologically in biopsies taken from individual people. So these are the direct effects of CMV. You have a named person where you can prove the virus is causing their disease. In contrast, CMV infection also has indirect effects, which you can define as an increased incidence of conditions that are also found in CMV seronegatives. So CMV isn't the cause of these, but it increases their incidence. And examples include atherosclerosis, immunosenescence, and mortality. So I'm going to discuss these in detail, but first, let's have an overview of the talk and the organisation. I'm going to start with 1 slide on a brief career outline, because that's what the organisers asked me to do. I'm going to show you a 1975 paper, which I believe is the first one to describe what we now call viral load. Looking around the audience, I suspect many of you weren't actually born in 1975. That makes me feel very old, but never mind. Then in 1982, I went to the Royal Free Campus, where we were doing a lot of organ transplants, and got some natural history data, and that worked up into a 1999 model of pathogenesis that I'll describe to you. I'll then talk about immune evasion and the host consequences of that, the indirect effects of CMV, a pharmacodynamic approach to vaccines, the lessons that we've learned from the glycoprotein B with MF59 adjuvant vaccine study in humans, and then consider future vaccines against CMV. So this brief career outline. So I went off to medical school, doing preclinical for 2 years to begin with, and then there was an option to do an extra year for an intercalated BSc, and I chose biochemistry because I wanted to do medical research and thought the techniques would be helpful to me in the future, and indeed they were. Then moved across to start the clinical year, and Professor Raymond Heath in the clinical introductory course gave a talk on virology. He said, I've only got an hour, but if anyone's interested, come over to the lab and we'll tell you a bit more about it. So I took up that option, got involved, and started on a project. In those days it was called Australia Antigen, again very old-fashioned, hepatitis B surface antigen it's now called. Started reading about CMV, got some projects going as a student there, and spent my final year elective in 1976 in Birmingham, Alabama, which is the centre of the universe for CMV research, and I'll tell you about that in a second. The current clinical problem at that time was rubella, and in the UK we were controlling that by diagnosing individual women and advising them to have terminations of pregnancy, and that was my job as a registrar, was to go and see these women and give them advice, and they were pretty upset, as you can imagine. I thought we've got to do much better than this. In the United States, they'd made vaccines against rubella, and that was controlling congenital rubella, a much better way of dealing with it. So, CMV damages more babies than does rubella with congenital infection, so why don't we just make a vaccine against CMV? Sounds easy, doesn't it? Right, well, this is the slide from 1975 that I discovered when I got to their lab. And what was happening is they were using the then fairly new technology of cell culture to diagnose infection in individual patients. We're seeing lots of kids with CMV, most of whom didn't have symptoms, which is quite unlike the situation with rubella. So in a remarkably prescient question, they asked, if it's not the presence of virus, might it be the quantity of virus they have? So every time they came back from the clinic with a sample from the children, they didn't just put them into cell cultures, they titrated them to look at what we would now call viral load. So they started off with children with perinatal CMV, shown in blue here, and this is clinically benign, and it's transmitted from mother to child through breastfeeding. So they have virus about 3 months of age. As a group, they have about this level of viral load. They looked at children who had asymptomatic congenital CMV, and as a group, they had about an extra log of viral load. And they looked at those with congenital CMV born with symptoms, and they had yet another log of viral load. And after 3 to 6 months, the curves became superimposable, but carried on with virus detectable for months into years. Now, the paediatricians were a bit disappointed with this slide because this is the standard error of the mean. There's a vast variation here, and they couldn't be certain that given a certain value, they were definitely in this group rather than this group, which is what they wanted to give advice to the parents. But for me, interested in viral pathogenesis, this was great. It said what you need to do is have a chemically defined assay that has much better precision and tells you, are you in this group or are you in that group, and allows you to follow up all the implications of CMV from that slide. Because that slide says that CMV is a systemic infection, so that sampling urine provides advice about clinically inaccessible sites such as the inner ear that you'd never be able to biopsy. CMV is a chronic infection in neonates, so there may be a CMV-specific immune defect. CMV may cause disease once a threshold value of viral load is exceeded, so treatment may be beneficial if the viral load is kept below this threshold value. And postnatal treatment may be beneficial even if it's for short term, covering the time when there's the difference between those curves. And remarkably, over the years, all those things have turned out to be true. Well, when I went back to London, we didn't have many cases of congenital CMV, but we did have the transplants. So along with Vince Emery, we set up an assay, quantitative competitive PCR, that allowed us to directly measure viral load in the same clinical sample, the same analytes, that is urine, and we can now express viral load in terms of genomes per mil of urine, a much more systematic analysis. And in following through prospectively renal transplant patients, we could then ask who got disease and how was this related to their viral load. And you can divide the groups into D plus R minus. This means the donor has antibodies to CMV and so is potentially infectious and transmits virus to a recipient who lacks immunity. Under those circumstances, with primary infection, they tend to get a high viral load, and some of them meet a case definition of CMV end organ disease. In contrast, these people reactivate their own virus, where the donor is irrelevant. They have a low viral load and a low risk of disease. And the group in the middle can either reactivate their own virus or become reinfected. It's some of the reinfections that get disease. So what is important here? Is it what's on the y-axis, the viral load, or is it what's on the x-axis, the immunity of the recipient? You need a multivariate statistical model. So univariately, using CMV end organ disease as the readout, viral load was significantly associated with end organ disease, as was viremia, and recipient positive serostatus was protective. The odds ratio was less than 1. But in the multivariate model, when you control for viral load, recipient serostatus disappeared as a protective factor, and viremia went from an impressively low p-value to one of borderline significance. But controlling for viremia and recipient positivity, the viral load remained essentially unchanged and remained significant. So it was this time, that was 1997, we concluded these patients were getting ill because they had a high viral load. And these other markers were useful clinically for making predictions simply because they were going to tell you who was about to have a high viral load. And to cut a long story short, the same was true when you measured the viral load in the blood of renal transplant patients or the blood of liver, bone marrow, and AIDS patients. When we looked in all the transplant patients and as a population of patients, plotted the probability of disease against their peak viral load, there's a non-linear relationship between them, the straw that breaks the camel's back, if you like, that we call the threshold concept. It's that extra log of viral load that causes the disease. And among those who receive steroids, those curves are shifted to the left by a median of about 0.3 of a log every time you have one treatment for a graft rejection. So we put all of those things into a multi-component model. From the time of transplant onwards, you start to produce virus. This may be in the transplanted organ, such as the kidney, or it may be in the salivary gland where latent CMV is present. There's some virus detectable, but it doesn't mean you'll get disease at that time, especially if the immune response can cope with the virus. But if it can't, it reaches a high level and spills over to give you viremia. Viremia is important because that's how the virus gets to different organs to give you retinitis, pneumonitis, gastrointestinal infection, etc. But it's not a guarantee of disease at those sites because the immune system locally may be able to control replication. But if it can't, it reaches a high viral load and triggers off a series of different pathological responses that end up giving you disease. All of the immunosuppressive drugs that the clinicians give to stop graft rejection work by putting up the viremia. So in a multivariate model, adding in doses of drugs doesn't actually give you anything else, except for steroids. And steroids work in this analogy by lowering the overflow, so you get disease at a lower viral load than would otherwise be the case. So what is important about this high viral load of CMV? Well, our working hypothesis is that a high viral load represents expression of the immune evasion genes of CMV. These allow CMV to persist in putative sanctuary sites of replication within the body. Short-term antiviral therapy can interfere with either the establishment or the maintenance of such sanctuary sites to reset the pathogenesis for that individual patient. And this therapy might provide clinical benefit. So, what are the immune evasion genes of CMV? Well, if you think of an immune defense, then CMV has countermeasures against them all. The first example here are cellular genes. These are the complement control proteins, and as the virus buds through the plasma membrane, it takes these proteins with it, so that if any complement tries to bind onto the virus particle, these degrade the complement. All the rest of these genes are viral encoded genes that map to the unique long, unique short, or the terminal repeat regions. There are at least 3 alpha chemokines and 4 receptors for beta chemokines. So you can see it's soaking up the beta chemokines from that local area and altering the balance between alpha and beta. There are many genes that interfere with interferon. There are several genes that are FC receptors, stopping the business end of antibody working. And there's an interleukin-10 gene. That's quite impressive to begin with, but there's more. On this cartoon, we're showing a virus-specific protein being synthesized on the ribosome, and it's targeted by ubiquitination for degradation in the proteasome. And the resulting peptides are transported through TAP to the lumen of the endoplasmic reticulum, where they associate with HLA class I molecules and go up through the trans-Golgi to the plasma membrane, where they display self plus non-self to T cells, come and kill this cell, it's virus infected, come and destroy it. If any of the HLA molecules are misfolded, they get re-exported back into the cytosol, and there's a healthy turnover of these cells. Specifically, the signal peptide from HLA molecules comes into the ER and upregulates HLA-E, and that provides a negative signal at the plasma membrane to say to natural killer cells, this is a healthy cell, it's got a healthy turnover, you don't need to attack it. So with that as background, how does CMV interfere with this process? Well, in as many ways as you can imagine. At the input virus level, PP65 and UL97 phosphorylate the major immediate early protein, such that it's less likely to be degraded in the proteasome and so less immunogenic. The protein from gene US6 sits here on the luminal side of TAP, blocking the transport of peptides. US3 sits in the ER and binds and sequesters HLA class I molecules. And US2 and US11 mimic this unfolded protein response and taking HLA molecules into the cytosol. These are regulated at immediate early and early times, so that before there's any DNA replication within the cell, the virus has taken over and blocked the ability of the displays that are required for cytotoxic T cells to see that this is a virus-infected cell. But what about that molecule, the HLA-E that I told you about? No problem. The virus has another gene, UL40, that produces a signal peptide that's identical to that of HLA class I molecules, and it goes up with HLA-E and provides the negative signal that the natural killer cells are looking for. There's another signal that macrophages are looking for with this particular receptor, no problem. the virus has a gene, UL18, that provides that signal. So these are two examples of decoy receptors, where the virus encodes molecules that actually send a negative signal to these white cells, telling them, nothing to see here, off you go and look somewhere else. The next stage of defense that the cell has are stress proteins that are released when the cell senses this unfolded protein response. And they send signals to natural killer cells saying that the cell has an abnormal turnover of HLA molecules and that natural killer cells should destroy the cell. And these are called the UL binding proteins because UL16 was the first example that was shown to block natural killer cells, and there have been several more of them. There's a whole list of them now. And if that weren't enough, the virus also encodes a microRNA that inhibits yet another member of the UL binding proteins. So the pattern you get is that this virus has multiple genes with overlapping functions that within this sanctuary site of this particular cell make sure that the immune system can't see the virus. It doesn't mind the fact that it's very fully immunogenic because the T cells can't actually find the virus-infected cells. But the T cells continue looking for virus, such that with time, they start to accumulate. And here's a healthy member of our laboratory staff, and we're using a tetramer to recognize a peptide that begins with NLV, the first three amino acids, from PB65, phosphoprotein 65, restricted by HLA-A2. And fully 3% of her CD8 cells are specific for one peptide of one protein of one virus. And these cells accumulate with time, and in elderly people, you find that they have more of these cells specific for CMV than they do to all the other viruses they've ever encountered in their life. And the idea, although it's controversial, is that this may give you disease in one of two general categories. The first is that these cells wish to attack CMV-infected cells but can't find them, so they attack something else instead, bystander attack, and can increase the incidence of conditions like atherosclerosis, which is an inflammatory condition. The second one is that because there are so many committed differentiated cells, there aren't enough naive cells to respond to new antigens. So in a topical example, you might take an elderly lady with lots of these cells, give her a vaccine against SARS coronavirus 2, she doesn't respond very well, she gets COVID and dies, and the doctor writes on the death certificate death due to COVID. Absolutely correct, but without recognizing that there may be an underlying feature. She may have survived if she hadn't had CMV underneath it. So these are the general ideas of how CMV may be increasing the incidence of some of these conditions, but evading our recognition of that contribution. So we should be doing something about those. And here is a small randomized controlled trial of valganciclovir given for 8 weeks compared to placebo, looking at the abundance of these T cells. And you can see that the abundance of cells comes down and stays down during a washout period afterwards. We should be doing more of this, particularly in the elderly people, and asking, if we follow them up for a long time, does that translate into a survival benefit? Because death is the endpoint that we're interested in here. And why death? Well, there's quite a bit of evidence that CMV is associated with an excess of death. This is a study from back in the early days of HIV. And if you presented with sight-threatening CMV retinitis at that time, the ophthalmologist's practice was to inject straight into your eye intravitreal ganciclovir, which actually, it sounds horrible, but actually was quite well tolerated by the patients and preserved their vision for a while. Some ophthalmologists, in addition to doing that, gave oral ganciclovir, so they had systemic therapy. And this group looked at all their patients from a cohort and said, do they do better if they have systemic therapy? And the answer was yes. If you look at the average point here, there's perhaps a 30 or 40% survival improvement if you had systemic anti-CMV therapy. There are other examples in the literature, but the best one comes from letermovir, CMV-specific inhibitor of the terminase complex. In the study in the New England Journal of Medicine, a randomized double-blind placebo-controlled trial, this reduced CMV end-organ disease as expected in stem cell transplant patients. But in addition, a secondary, pre-specified secondary endpoint was all-cause mortality, and that was reduced as well. And this reanalysis by Lungman and colleagues, on the left-hand panel, you see the group of people who at any point had CMV viremia, where you can see a survival benefit from the use of letermovir. And here are the people who never had CMV viremia, where there's no difference. So it isn't that letermovir is a drug that's going to increase the life expectancy of anybody. It's a drug that takes out the association of CMV viremia with excess mortality and returns it back to where it should be. So this is the first proven example of an indirect effect of CMV. Unequivocally, CMV is the cause of excess death in the stem cell transplant patients. There's also an excess of death in the general population. You'd much rather be following this curve than this curve, I'm sure, in follow-up with time. The data come from the NHANES study. And the CMV association I've described to you there was attenuated by diabetes, by obesity, and smoking at baseline, but remained significant after they were controlled. The population had 14,000 people from the USA, 13.7 years of follow-up, with a hazard ratio of 1.19, a 19% increase in mortality, quite a large amount. And another group with a large population tried to replicate their results and came up with very similar results. In the UK, 13,000 people, 14.3 years of follow-up, hazard ratio 1.16. So it's definitely consistent and reproducible. What would you say about overall the indirect effects of CMV? That association with mortality may not be causal. It hasn't been proven to be causal. But at the same time, there could be unrecognized confounding, for example. But at the same time, there is a case to answer, and it's obviously biologically important to get to the bottom of whether there is a mortality difference, especially now Latoma's has been shown to inhibit mortality, reduce mortality in stem cell transplant patients. So we need more placebo-controlled trials of antivirals to provide evidence for or against causality. And when we get to phase 3 randomized controlled trials of prototype vaccines, I think we should incorporate long-term follow-up for mortality, because if the vaccines were to improve mortality, that would improve their cost effectiveness even more and make them much more popular with the customer who's being offered the vaccine. These vaccines are already predicted to be cost effective. If they really could reduce the indirect effects of CMV, they would be cost saving, which would be remarkable. And there's a problem. How would a public health planner deciding whether or not to invest in making the next prototype for CMV vaccine, how would they take on board that information? Should you just believe it all and say, oh yes, CMV will definitely reduce mortality? Or should you say, no, I don't believe a word of it, there's unrecognized confounding? There is a middle point. What weights would you attach to those figures? And I've tried to do this in this article, looking at the probability of public health benefit from universal immunization. And we're going to look at several different things. HIV, atherosclerosis. I haven't told you, but on intensive care, patients admitted to intensive care, there's an excess of duration of ventilation in those who are CMV positive. In the elderly, the immunosenescence, I've shown you a bit about that, and in the general population, mortality. If you have a study with a plausible, reproducible natural history from several different investigators, I would say, well, perhaps 10% of that potential cost, I would say, perhaps could be returned if we had a vaccine. You can go ahead and invest on that particular basis. If there was an encouraging antiviral randomized controlled trial with a surrogate marker, then perhaps I'd accept 25% overall. And if you had an encouraging antiviral randomized controlled trial with clinical endpoint, then perhaps 50% of the cost. You'd never accept 100% because until it's finally proven. But you could put this into your planning purposes. And I've chosen those because we already have some evidence from some of them already. All of these things and a couple of those over there. And frankly, anything that can reduce the days you spend in intensive care because it is so expensive would likely be cost saving. So these figures should encourage vaccine manufacturers to develop their vaccines and bring them forward and to incorporate mortality as a long-term potential benefit. These figures, of course, purely arbitrary. You can choose your own figures and play with the numbers. So all we need now is a vaccine. Where are we with a vaccine? Well, to give you a balanced talk, because if you haven't caught it yet, I'm an enthusiast for CMV vaccines. I need to give you one slide on pessimism, because these are the sorts of comments that over the decades I've had from grant reviewers saying, quotes, it'll be impossible to make a vaccine against CMV. They raise some reasonable points. They say things, it's a large genome, it encodes for 170 genes, all the proteins are fully immunogenic, as shown by this heroic work by Sylvester and colleagues. The immune system is already committing a lot of resources to this virus. CMV also has multiple immune evasion genes, so whatever you induce, the virus will escape from them. Immune individuals can be reinfected. This isn't a pattern. This isn't propitious for making a new vaccine. We should go for something simple, like measles. There's also a direct correlation between the proportion of women who are seropositive in a community and the incidence of congenital infection, as shown here with a paper from the CDC. As you get up to 100% of seropositive women, the incidence of congenital infection increases. With rubella, as you get up to 100% of women who are immune, the infection comes down to 0. So people say, there you are, nobody understands CMV, it's too complex, and if you give everybody extra immunity, you may make things worse. Don't even bother to try. So I accept that a successful vaccine would have to do better than nature. It's just that I don't think it will be difficult to achieve that, because this isn't a fair comparison of what the immune system can do. It is true that 100% of these women are seropositive, but it's also true that 100% of them have latent CMV that can reactivate during pregnancy and transmit to their fetus, which is the explanation for why this number goes up at the end. It's also true that these are 100% of women who've had their immune system, immune response to CMV induced in the presence of the immune evasion genes, because they got their immunity from infection from a virus that arguably wants us to have this impaired immune system so it can carry on reactivating. It's not a fair comparison, assessment of what a vaccine can do. What we need to do that, we need to go and give people immunogens that make them make good responses against CMV, hopefully with a good adjuvant as well. And under those circumstances, you can imagine, on the most optimistic scenario, that you might be able to prevent initial primary infection. If you can prevent primary infection, you can prevent the establishment of latency. And if you don't have latent virus infection, you can't be reinfected. So all of this complex natural history could all disappear if only we had a really good vaccine at preventing primary infection to begin with. Now, I'm told that that is naive and simplistic and overly optimistic, and it is too optimistic. But we should set ourselves that target of trying to aim for it, keep bringing forward more vaccines and seeing how close do they get to this ideal. And I would point out that over the years, another example, an analogy has come through with hepatitis B vaccine. That's a virus that persists within individuals and causes long-term disease, but can be prevented by simple immunization with a simple glycoprotein vaccine. So we shouldn't ignore the possibility that might work for CMV as well. Okay, so we need to go on and develop vaccines, but how are we going to do it? How are we going to show that people are protected when this is a virus that doesn't usually cause any symptoms? Well, this is where our transplant patients can help out. CMV is such an important problem that transplant centers have protocols for handling it. And the antiviral drugs are given either prophylactically here for a fixed period of time when they're effective at suppressing clinical symptoms, but some people come back with late onset disease, often with resistant strains. Or you don't give the drug to anyone to begin with, you monitor everybody, you ignore the low viral loads, remember the threshold concept, we're not interested in low viral loads. When they get a moderate viral load, you treat them until they become negative, that person becomes negative, personalized medicine, follow them, and if they have a second episode, treat them again. And under those circumstances, we have a low incidence of end organ disease, and we don't have late onset disease. Importantly, this gives us all of these parameters of viral load and duration, and we can use that as our model. All we have to do now is give vaccine or placebo pre-transplant and ask, can we alter these viral load parameters? So what happens to the natural history? This is a study from a decade ago now, where we went live with real-time quantitative PCR to monitor the transplant patients, and they were renal transplants or liver transplants. There were 689 of them. They're divided into 4 groups as to whether the donor or the recipient, or neither or both, has IgG antibodies against the virus at baseline. And you can see, for example, the high-risk patients are the D+R- at risk of primary infection. There are only 11% of patients, but 20% of those with viremia and 36% of those with high-level viremia who need to be treated in order to prevent disease. So you can use this to ask simple questions about natural history. How often does the virus transmit in a primary infection when the donor is positive and the recipient is negative? 78%. How often do people reactivate their own virus? 40%. How often do people become reinfected? Well, if you accept the argument that these guys have the same risk of 40% of reactivation, then the difference between them, the extra 14%, must be due to reinfection. Or to put that another way, a positive donor transmits to 78% of people without immunity, but only 14% of people with immunity. So natural immunity has some activity, it's just not perfect. And who gets the high viral loads? Well, it's nearly everybody in this group, a minority here, and an intermediate value for those guys. The other biomarkers you can use are the duration of viremia, that's significantly different across these 3 groups in the 2 patient populations, and peak viral load, which approximates to a normal distribution in the D+R-, but is shifted dramatically to the left, this is a log scale, in the D+R+. The difference between these 2 groups is they both have a positive donor, but here the recipient has no immunity, and here the recipient has natural immunity. So natural immunity is quite good at protecting you against a high viral load. It's just not very good at protecting you against a low viral load, any more than it can protect you against a low viral load associated with reactivation of virus. This is a herpes virus, after all. So what we want to do is give vaccine or placebo to these people pre-transplant and ask, can we alter this distribution. So we want preemptive therapy applied to CMV vaccine discovery. Could vaccine-induced immunity protect against a high viral load in transplant patients in the way that natural immunity does? And should that be cellular or humoral? And how potent would a vaccine need to be? Would you need to go for sterilizing immunity that you might define as the inability to detect virus or a component of virus in any body fluid at any time after transplant, a very difficult target? Or would it simply be sufficient to prevent the threshold effect that I've shown you already? And our assessment is that if you could shift this curve to the left by just over a log or so, you could potentially abolish disease, although they would still all have infection. To put that in a contemporary example, you might have a lot of people who have a vaccine that stops them getting COVID, but they still all have SARS coronavirus infection. Who cares? So we decided to proceed to a proof of concept study using the first vaccine to become available, glycoprotein B together with MF59 adjuvant, kindly provided by Sanofi, that had completed its phase 1 clinical safety studies. So we took 70 seronegative recipients who were awaiting transplantation, solid organ transplantation, gave them vaccine or placebo 0, 1 and 6 months, and afterwards, when the code was broken, looked at their geometric mean antibody titer. The antibody negative, seronegatives remain seronegative. The seropositives have a significant increase in antibodies, which declines a bit, but after the third dose comes up to a level that's about the same, as I'll show you on the next slide, as natural seropositives. So our first conclusion is this simple vaccine can give you as much immunity against glycoprotein B as you get from natural infection. The levels decline but when they come through to the time of transplant, but are still significantly different at that time. In the seropositives, their antibody titer remains fairly constant, but if you give with placebo, but if you give vaccine, you have about a half a log increase in their antibody level, and there's still a difference at the time of transplant. So our second conclusion is we can do better than nature. We can give you more antibody against glycoprotein B than you would normally have. But does it matter? Does it control virus replication? Well, you can read the details in this paper from years ago now. But yes, the vaccine reduced viral load. It reduced transmission from donor to recipient, and the correlate of immunity was the titer of antibodies made against the glycoprotein B that was in the vaccine. So the virus encodes 170 proteins, and our focus has been reduced towards one of them, glycoprotein B. It doesn't mean it's the only component of a future vaccine, but it's certainly something that people should consider when they're developing a new vaccine. And we've subsequently been interested in epitope-specific responses to this protein. This was helped by Burke and colleagues who published the crystal structure. And you can see in this monomer, there are extensive regions of alpha helix and also of beta sheet. That's a monomer, and this is the trimer, and this is the space-filling version of the trimer. And immunologists have studied in great detail the B cells that come from seropositive individuals and asked which antigenic domains do they recognize on this protein. Antigenic domain 1, AD1, is immunodominant. 100% of seropositive people have antibodies against that particular domain. It's a linear epitope, and most of those responses are non-neutralizing. Antigenic domain 2 is also a linear determinant, but many of those, it's only found in 50% of the population, but many of those are neutralizing. Antigenic domain 3 is a bit strange. It's on the transmembrane or intracytoplasmic or intraviral part of the molecules. It's quite difficult to think how antibodies, once they're made, would actually reach that target, but nevertheless it is immunogenic. That's a linear peptide. And AD4 is a discontinuous epitope in this green part of the molecule, and AD5 is a discontinuous epitope in the blue part of the molecule. So importantly, not just the immunology, but which is protective. So our immunology colleagues were saying to us, oh, just immunize people with AD1. If it's immunodominant, that's the thing to go for. And I'm saying, no, I suspect that's a decoy put there by the virus. We should be looking at some of the other ones. They could easily be the ones that are protective. So we had a bright PhD student, Ilona Baraniak, who published a series of different papers. In seropositives, AD1 is not protective. It's AD2 that correlates with protection, the one that's found in only 50% of people. Your prior experience as a seropositive of CMV antigens directs how you will respond to the vaccine, the concept of original antigenic sin as originally described for influenza. In the seronegatives, protection was definitely not mediated by neutralization or antibody-dependent cellular cytotoxicity measured at the time that they were immunized. However, the transplant that they subsequently get acts like a prime boost immunologically, including a boost to their neutralizing antibodies that come up later. We also wanted to prove really that humoral immunity was the important component, was actually the mechanism that explained what was happening. And the experiment you want to do is quite obvious here. You take a seronegative recipient who's waiting for a transplant and he's destined to receive one from a seropositive individual, and you invite them to enter into a randomized controlled trial where they receive either monoclonal antibodies specific to CMV or a matching placebo just at the time that we're transplanting the kidney. And if you can show that you reduce transmission from donor to recipient under those circumstances, then you know the quality of monoclonal antibodies that must be present, and you know the quantity, because you're taking serial bloods from these people, and you can put those two things together and understand what's going on in what is effectively a human challenge model. So I proposed this to Genentech, who have these antibodies, and unfortunately they didn't have a clinical grade anti-GB antibody, but they did have a couple of others. One against glycoprotein H, and GH together with GL and GO bind onto a receptor on fibroblasts, whereas GH, GL together with three other proteins bind on, they form a pentamer, and they bind onto a receptor on epithelial and endothelial cells. So they had two monoclonal antibodies, one against GH and one against this component of the pentameric complex. Some patients received that, and some patients were randomized to receive placebo. And these proteins are just upstream of the fusion function of glycoprotein B that they interact with after infection. And what happened? There was a significant reduction in transmission hazard ratio of 0.5 from donor to recipient. So it is proven that it's humoral immunity that interrupts transmission from donor to recipient, and we need to try to improve the efficacy of this approach. That same vaccine also protects women. Bob Pass did a nice study in seronegative women who just had a baby. They received 3 doses of glycoprotein B with MF59 or a matching placebo. They used the same preparation, the same schedule as I've already told you for the solid organ transplants. Importantly, the correlates of protection were similar, meaning that what we do in transplants can be applied to women of childbearing age. The protection definitely was not conventional neutralization, and responses to AD2 and AD3 were important, and these papers from Sally Permar's lab includes a whole series of new techniques, including how the antigen might be presented, which can help us design more sophisticated analyses than are usually done in evaluating vaccines. So I'd guide you towards those papers. So what are going to be the next steps? Well, I think clinical trials will define how to control CMV in selected populations. They will define the minimal number of immunogens and tell us whether humoral or cell-mediated immunity or both are required. They will aim to induce protective immune responses against glycoprotein B, but also against PP65, which is the major target of cell-mediated immunity, and also the pentameric proteins, inducing both humoral and cellular immunity. They will try to avoid immune evasion and also the decoys, multiple ones that I've described to you already, but including now glycoprotein B AD1. So you see the problem immediately. You want to induce protective responses against GB, but you want to avoid the decoy that's also found in that molecule. So a better understanding of what's going on can help us design the next range of vaccine studies. So far, we've used what you might call first-generation vaccines, the original town live attenuated vaccine studied by Stanley Plotkin, plasmids that presented PP65 together with glycoprotein B, and these predominantly induced cell-mediated type responses because the GB response was quite poor. And there's the recombinant GB and MF59 that predominantly induces humoral immunity. Second-generation vaccines you can define as those that induce both humoral and cell-mediated immunity, and there are 3 of them in clinical trial at the moment. Merck have a whole virion vaccine, which is being studied in women of childbearing age. A company called HooKeeper have glycoprotein B plus a second construct containing PP65 expressed in lymphocytic choriomeningitis virus, and that's in transplant patients. And Moderna are using their RNAs to produce GB plus the pentamer proteins, also in women of childbearing years. Mostly seronegative, but some seropositive. These studies should come out, we should get full results in the next 12 months, and this one is about a year or so behind that. So within a couple of years, we will know whether the hypothesis that giving generous amounts of immune response, because the phase 1 studies show that essentially you get, at least in healthy volunteers, you get the sort of, at least the sort of a level that you find of cell-mediated and humoral immunity in natural seropositives, whether that is sufficient to control CMV replication in communities. And then hopefully one or more of them will come up with positive results and they will be licensed, and then we can deploy them for communities. But we need to be aware that CMV may be more tricky than that. We may need to think of other things. Will we need a third generation of vaccines? And the definition of third generation vaccines might be, let's express the cryptic antigens that are not normally presented by CMV because it doesn't want us to see them. It, as it were, knows, if you let me personify the virus, that these would prevent its infection, so it keeps them hidden. We need to try and discover those and try to put those into vaccines and then ask, do they provide additional clinical benefit over and above these? And if you want an example of one of those examples, then Matt Reeves is going to give a talk tomorrow on a potential candidate in that area. So my time is up, and I'm going to end with acknowledgements to all of our clinical colleagues, liver transplant, renal transplant, and bone marrow team who've let us study their populations of patients over decades. The patients themselves who volunteered for all of these studies, Emily, who's recruited them into the studies, my colleagues Vince and Matt for great scientific work, Claire and the people in the diagnostic lab, Colette, who keeps us honest with the interpretation of statistics, and the Wellcome Trust, who I must say have funded me since I was a student and then through multiple project grants and multiple program grants as I got very old. And finally, the National Institutes of Health, who supported us for doing the vaccine study in the transplant patients. Thank you.
I'd like to thank Paul for what was an outstanding and fascinating lecture for the Unilever Colworth Prize. And on behalf of us all, I'd like to present him with his certificate.